In Spain, the implant market is worth over 200 million euros, with around 1.5 million implants sold. Higher life expectancy among the general population is one of the key indicators which will consolidate the future growth and importance of this sector. Given this scenario, it is necessary to endeavour to assure the continued improvement in the results of implantology treatments, while considering both the manufacturer and the patient. Researchers from the Polymers and Advanced Materials group at the Universitat Jaume I of Castelló have developed an in vitro method to predict the capability of an implant to induce soft and bone tissue regeneration when in contact with those tissues. This may apply to hip, knee and dental implants, transepithelial abutments, etc. The first element of evidence taken into account is that there is a poor correlation between in vitro tests and subsequent in vivo experiments. Results obtained in the lab do not respond in the same way when applied to real cases. This implies an enormous waste of research time, significant investments, uncertainty when selecting a prototype, and a slow development of new healthcare products. How can this problem be tackled? Here is where proteomics comes into play. Our blood contains many proteins that come into contact with an implant when it is placed in our body. As a result, these proteins adhere in different ways to its surface and consequently form a layer. Depending on the type of implant and its properties, a different protein layer is created, and depending on the layer composition, different biological responses can be observed. Therefore, protein absorption onto the implant determines whether the regeneration will be successful or not. The systematic proteomic study of biomaterials with different biological responses has enabled the identification of the key biomarkers that will allow for the prediction of how an implant will behave in our body. Based on these biomarkers, a new technique is developed to determine the potential of an implant to regenerate different types of tissue more efficiently. Thus, for example, with a new dental implant, the researchers would try to establish its osseointegration capability, and when developing a transepithelial abutment, they will try to determine the capability of the soft tissue to seal around this particular implant. This research project can be applied not only to dental implants, but also to any type of prosthesis. So it can be very useful to those who manufacture materials that must be in contact with tissues. Definitively, we can welcome a new method that is safer, faster, and more economical, as it will reduce costs for implant manufacturers. When putting a new product on the market, this innovation will minimize risks for patients, as well as decreasing testing times and saving costs.